of the day. This is a combination of a couple of different areas. We'll start off with a talk on gene therapy, just to broach the topic of new therapeutics. And I'm delighted that Lei Xu will be joining us to give that talk. That'll be followed by Lianne Hu's talk on CAR T therapy, something that's extremely topical and very promising in many previously untreatable diseases. And finally, just a broad overview of international clinical trials from Kassa Ayalew. Kassa will tell us about the importance of international data and how important a role it plays in virtually all the studies that we look at. So I think at this point, we're ready to turn over to Lei to begin discussing gene therapy.
Thank you, Dr. Sachs, for the kind introduction. I also would like to thank you all for being here. I hope after my talk, you will have a better understanding of important principles on efficient clinical development of investigational gene therapy products, a better understanding of regulatory requirement and flexibility for marketing approval of these products, and finally, a better understanding of toxicities associated with adeno-associated viral vector-based gene therapy products. My talk will focus on the in vivo gene therapy products. Those are products delivering genetic material by direct injection into patient using viral or non-viral vectors. One of the common viral vectors is adeno-associated viral vector. Since 2017, FDA has approved 3 in vivo gene therapy products. All of them are AAV-based. The most recent one was approved last week, and it is indicated for adults with hemophilia. There are similar fundamental considerations regarding the clinical development between gene therapy products and other biological products. The clinical development programs for each product for different conditions may vary substantially. So we encourage sponsors discuss with us early in the clinical development using the meetings such as Interact and the pre-IND meeting formats. For early phase trials, the safety is the primary objective, but because usually these trials enroll patients with the conditions of interest, it is possible to also obtain information on activity and the preliminary clinical efficacy if it is appropriately designed. We encourage sponsors to try to hit a home run, which means the design of the first in-human studies will be able to provide evidence of effectiveness. In order for the trials to be able to provide evidence of effectiveness, it is important to consider the design elements such as randomization, concurrent control, and blinding. And also, the sponsor should resolve as many manufacturing issues as possible before the initiation of the first clinical trial. We believe randomized concurrent controlled clinical trials with appropriate blinding may be the most efficient means of obtaining persuasive evidence of effectiveness. Many diseases have poorly understood etiology or pathophysiology, are poorly characterized, or have highly variable natural history. All this make the use of external control very challenging. We encourage sponsors to consider randomized concurrent controlled double-blinded clinical trials, even for the first in-human studies, because this would facilitate data interpretation, and this is especially important for rare diseases because it can not only maximize the use of valuable patient resources, but also may provide sufficient evidence of effectiveness to support a future marketing application. And all subjects should receive standard of care and then be randomized to the added gene therapy product or a control, such as a placebo. External historical control may be appropriate if all criteria are met. That is, an unmet medical need. It is not practical to have a concurrent control. The disease cause is well documented and highly predictable. The study population and historical controls are suitably comparable, and the treatment effect would be large and self-evident following the product administration. In most cases, the historical controls may be inadequate because it is very difficult to meet all the criteria listed above. So in general, the use of external historical control in place of a concurrent control is not encouraged. For later phase trials that are intended to provide the primary evidence of the effectiveness, the primary efficacy endpoints should be either clinically meaningful endpoints, which directly measure how patients feel, function, or survive, or surrogate endpoints that are reasonably likely to predict clinical benefit. And for the regulatory approval, the requirement is that it has to be based on substantial evidence of effectiveness and evidence of safety. And the evidence of effectiveness should be obtained from adequate and well-controlled studies. We understand that certain aspects of product development that are feasible for common conditions may not be feasible for rare diseases. So FDA's regulations provide flexibility in applying regulatory standards. For example, for rare diseases, there is no specific minimal number of patients to be studied to establish the substantial evidence of effectiveness and safety. And the number of the patients to establish such evidence is determined on a case-by-case basis, taking several factors into consideration, such as the persuasiveness of the data, the nature of the benefit, the patient population that will be treated after approval, and the safety profile. In general, to obtain substantial evidence of effectiveness, we require more than one adequate and well-controlled studies because the consistency of results across two adequate, well-controlled trials greatly reduces the possibility that a biased, chance, site-specific, or fraudulent results will lead to an erroneous conclusion that a product is effective. However, under certain circumstances, we can conclude that one adequate, well-controlled clinical investigation plus confirmatory evidence is sufficient to establish the substantial evidence of effectiveness for a marketing approval. And a couple of factors are considered, including the persuasiveness of the single trial, the robustness of the confirmatory evidence, the seriousness of the condition, the size of the patient population, and whether it is ethical and practical to conduct more than one adequate and well-controlled clinical investigation. Now I would like to use a Brach-Nipovac as an example of how we apply the regulatory requirement and regulatory flexibility. This is a first-in-class AAV-based gene therapy product delivered subretinally, and for the indication of confirmed biallelic RPE65 mutation-associated retinal dystrophy. This is a rare condition with about 1,000 to 2,000 patients in the US. Patients usually start with night blindness and progresses to visual field loss and eventually complete blindness. It has a significant impact on their activity of daily living. There was no approved pharmacological treatment prior to the approval of this product. And the picture on the left upper corner shows people with normal vision when they see the world during the late afternoon. And the picture on the right shows the patients with the condition and how they see that world because of the decreased light sensitivity. So to assess the clinical benefit of this product for this patient population, sponsor and FDA worked closely to develop this novel endpoint, multiluminance mobility test, which assess the patient's ability to accurately mobilize at different light levels. And this endpoint was eventually used in the only phase 3 trial that provided the primary evidence of effectiveness. So in summary, in this program, we used a novel endpoint. There is also non-traditional clinical development program because they have one phase 1 study and followed by a phase 3 study. There is no phase 2 study in between. And also this product received both breakthrough and the priority review designations. So lastly, I'm going to talk about the toxicities associated with AAV-based gene therapy products. Hepatotoxicity, hematological toxicity, and the neurological toxicities have been observed in AAV-based gene therapy clinical trials. In the next several slides, I will focus more on the hepatotoxicity, thrombotic microangiopathy, and brain MRI abnormality. Hepatotoxicity is the most common adverse event associated with intravenous or systemic administration of AAV-based gene therapy products. Prophylactic use of corticosteroids seem to somewhat mitigate the risk of hepatotoxicity. Hepatotoxicity can present in a variety of severities. The most common one is the elevated liver enzymes, but patients are asymptomatic otherwise. It can also present as drug-induced liver injury as defined by Hy's law. In more severe cases, hepatic failure, even death, can occur. It is speculated that anti-AAV T cells may play a role in the development of hepatotoxicity. And this is why corticosteroids, which can inhibit these T cells, can mitigate the hepatotoxicity. About 1/3 of SMA clinical trial participants had at least one adverse event of hepatotoxicity. There were cases of elevated aminotransferases over 20 times of upper limit of normal and had to be treated with corticosteroids. There was also one case of acute serious liver injury or liver failure. The patient developed jaundice, elevated total bilirubin, prolonged prothrombin time, about 7 weeks after treatment. Liver biopsy showed massive hepatocyte degeneration and inflammatory infiltrates. The patient recovered following the corticosteroids treatment. The risk mitigation for the hepatotoxicity included the box warning and the recommended systemic corticosteroids used in the U.S. prescribing information. There were also additional post-marketing cases reported regarding the hepatotoxicity. Next, I'm going to talk about thrombotic microangiopathy, or TMA. It is a hematological emergency. It is due to damage to arterioles and capillaries and macrovascular thrombosis. It is a syndrome of hemolytic anemia, thrombocytopenia, and acute kidney injury. There are two primary forms, thrombotic thrombocytopenic purpura and hemolytic uremic syndrome. Several cases of thrombotic microangiopathy were reported after receiving onisaminagene abelparvovec. The clinical manifestations usually started about 1 week after receiving the product. All those patients received the recommended prophylactic dose of prednisolone. It is unclear whether there was a role of concomitant of triggering events, such as infection or immunization. There was laboratory evidence of complement activation. Majority of those patients recovered from thrombotic microangiopathy within 2 to 4 weeks. However, there was one patient who had persistent hypertension, and another patient had hypertension and nephrotic syndrome lasted for about 3 months. All these cases of TMA occurred post-approval. As a result, the USPI was subsequently updated to include thrombotic microangiopathy under Section 5, warning and precaution. The last is neurotoxicity, which is the brain MRI abnormalities. The direct intraparenchymal delivery of AAV vectors to specific parts of brain is attractive for the diseases affecting central nervous system because it may improve targeting efficacy, avoiding administering high systemic doses of AAV to cross blood-brain barrier, and also to decrease the risk of systemic toxicity of the high dose of AAV vector. In an early phase clinical trial enrolling patients with late infantile Baden's disease, brain MRI T2 hyperintensities associated with intraparenchymal administration of AAV Rh10 vector were observed in all 13 patients 48 hours after the product administration. The hyperintensity findings were localized to the sites of administration. The findings were persistent in 7 participants after 18 months, but was resolved in 2 participants. In addition, serious adverse events during the acute period were observed in 6 participants. However, it is difficult to determine the causes of these serious events, whether it is due to AAV vector, the delivery procedure, versus the delivery device. In summary, an early phase clinical trial of a gene therapy product for a rare disorder may provide evidence of effectiveness and safety. We encourage randomized concurrent controlled early phase trial with appropriate blinding. We encourage the sponsors to resolve the product's manufacturing issues as much as possible before initiating the first in-human clinical trial. Approval of gene therapy products for rare or for common diseases must be based on substantial evidence of effectiveness and evidence of safety. Evidence of effectiveness should be obtained from adequate and well-controlled clinical trials. Serious and life-threatening toxicities have been reported as more and more patients receive AAV-based gene therapy products. Now it's time for challenging questions. The first question is, a concurrently controlled, randomized first-in-human early phase study is not necessary or recommended because the objective of such a study is to assess safety. True or false? I will wait for a second. Yes, this is false. We encourage sponsors to conduct randomized, concurrent controlled early phase study as soon as possible, even for the first in-human study. And this is particularly important for products for rare conditions. So question 2, which of the following is the most common toxicity of the AAV-based gene therapy products? Oncogenicity, hepatotoxicity, thrombotic microangiopathy, or neurotoxicity? Yes, you are correct. Hepatotoxicity is the most common toxicities observed for the AAV-based products. So this comes to an end of my presentation. Thank you for your attention. I will come back to address your questions during the Q&A session a little bit later.
Welcome to the Clinical Investigator Training course. In this session, we will provide a brief review of clinical considerations for the development of chimeric antigen receptor, also known as CAR, T-cell therapy for cancer. My name is Lianne Hu. I'm a clinical reviewer in oncology branch at CBER, which stands for Center for Biologics Evaluation and Research. This review includes opinions from me and my colleagues and do not represent FDA policies. I have no conflict of interest to disclose. In February of this year, FDA approved CARVYKTI for refractory multiple myeloma, which marks the 6th FDA-approved CAR T-cells. As we understand, most of these approved CAR T therapies are indicated for patients with a B-cell malignancy and who have already had 2 or more prior systemic therapies, and approvals were mostly based on single-arm trials with high objective response rate ranging from 50s to 90% and high complete response rate ranging from 30s to 60%. I would like to highlight 2 recent approvals for early treatment line. As noted in red on this slide, in 2022, FDA approved Yescarta in the second-line setting based on results from a randomized control trial, and Breyanzi in the second-line setting based on combined results from a randomized control trial and a single-arm trial. These approvals mark the expansion of CAR T-cell therapies into earlier lines of cancer treatment. However, the clinical development of CAR therapies in solid tumors has not led to an approval. This field still faces many challenges, including intolerable toxicities and low efficacies. Most of the CAR development in solid tumors are still in early phases. Therefore, our review today will focus on clinical considerations for early phase trials. We have 3 learning objectives. After this short review, you should know what are the major clinical considerations in early phase clinical development of CAR products. Second, common safety information FDA seeks from sponsors. Third, maximum tolerated dose may be different from optimal dose. Just to note, at the end of this talk, I will have 2 challenge questions to test your knowledge about the learning objectives. These are the outlines of this brief review. First is a brief introduction about how oncology products are regulated and reviewed at the FDA. Then the main review of today is to highlight some of the important clinical considerations. At the end of this talk, we will have a brief introduction of meetings between sponsors and FDA. Oncology products are regulated by 3 different centers at the FDA, which are CDER, CBER, and CDRH. In addition, OCE, which stands for Oncology Center of Excellence, which is under CDER, but it oversees the development of all oncology products, including biologics, regulated by CBER, especially in the late stage of the clinical development. Review at the FDA involves a multidisciplinary team. Each team has its distinct role and responsibilities, but discussions among us take place whenever it's needed. We're now having more than 270 research INDs involving a CAR product. Most of them are CAR T cells, a few are CAR NK cells, and CAR macrophages. Almost all are for cancer treatment development. 64% of these INDs are for hematologic malignancies and 36 for solid tumors. 86 of these products are autologous CARs and 14% of them are allogeneic CARs. CAR T cells are living T cells produced either from patients, so-called autologous CAR, or healthy donors, so-called allogeneic CAR. In general, CAR construct is engineered into T cells using retrovirus vectors. CAR T cells usually target cell surface antigens and are not restricted by HLA typing. This cartoon depicts the evolution of generations of CAR T cells. The basic component of a CAR include an antigen binding domain, a hinge, a transmembrane domain, and an intracellular signaling domain. Since the first generation of CAR T cells was developed in 1993, 4 generations of CAR T cells have been developed. The 5th generation is actively under development. Much effort has been on enhancing safety, persistence, proliferation, efficacy using genetic engineering techniques, including genome editing, especially CRISPR techniques. Now, I would like to give a brief review of some of the key clinical considerations for early phase trials of CAR products. I would like to note that in March of this year, FDA published 2 draft guidance. One is specific for CAR products. The other one is for gene products incorporating human genome editing. Please refer to these 2 guidance in the guidance section at the end of this slide deck. The first clinical consideration is trial objectives. For first-in-human studies, the primary objective should always be safety and feasibility. Usually, a dose escalation study is designed to identify maximum tolerated dose, MTD. However, in the era of molecularly targeted therapies, more and more evidence suggests that MTD from small dose escalation studies may be far from optimal. For example, dose-response relationships for CAR T therapies are often unclear. The expansion and persistence of CAR T cells depend on many intrinsic and extrinsic factors, and long-term toxicities of immunotherapies are unpredictable. For these reasons, we often recommend that sponsors evaluate at least 2 different dose levels in the dose expansion studies. In terms of eligibility criteria, I would like to highlight a couple of key points. First, early phase CAR T trials are usually conducted in patients whose disease have progressed on standard systemic therapies and who have very few or no treatment options otherwise. Sponsors should ensure that key inclusion and exclusion criteria are very clear, not too broad. Risks to the intended study population should be justified by the potential benefits. Second, if an inclusion criterion involves an assay of a specific antigen, the assay is likely to be considered in vitro companion diagnostic. In this case, the FDA will assess whether in vitro diagnostic poses a significant risk to the intended study population. We generally ask sponsors to provide an initial assessment of significant risk or non-significant risk and submit it along with their IND application. Now, I would like to highlight several considerations in study design endpoints and confounders. First, early phase trials of CAR products are generally open-label single-arm trials intended to evaluate safety, tolerability, and feasibility. Preliminary anti-tumor activities assessed by objective response rate and duration of response is critical to justify risks to the intended study population and also to identify the optimal dose. Second, objective response means either complete response or partial response, but not a stable disease, because it's challenging to interpret stable disease in the single-arm setting. For the similar reason, single-arm trial design is not appropriate design for studying time to event, such as progression-free survival and overall survival. Third, sponsors should consider important confounding factors which could affect the interpretation of future study results. Lastly, we suggest that sponsors consider randomized control trial design as early in the clinical development program as possible if the proposed treatment is a combination therapy. Next, let's talk about starting dose and dose escalation studies. Starting dose of a proposed CAR T therapy may be based on toxicology data from preclinical data, but we may accept a proposed starting dose based on a safe dose of another CAR product with same or similar target antigen or similar CAR structure, as long as the justifications are reasonable. To calculate dose level, FDA generally recommends the number of transduced cells. This is because gene transduction frequency may differ from lot to lot, and the best way to reduce those variations among study subjects within the same dose cohort is to use the number of transduced cells. The dose can be based on per body weight, per body surface area, or flat dose. For dose escalation scheme, most common scheme is 3+3 design. However, more and more sponsors use model-based design, which may be more efficient and accurate in identifying MTD and optimal dose. For dose escalation, we usually recommend half a log increment for CAR products. Depending on the product, we commonly recommend at least 14 to 28 days of staggering among the first several study subjects in each dose cohort. Properly identified DLTs is a key to dose escalation and the successful identification of MTD. The rule of thumb is that grade 3 or higher non-hematologic toxicities and grade 4 or higher hematologic toxicities should be defined as DLTs, with exceptions. We review sponsors' proposed exceptions carefully, taking into account characteristics of the product, study subjects' underlying disease stages, metastasis status, and their general health conditions. We commonly recommend at least 28 days of window for evaluation of DLTs following the infusion of CAR T cells. As the most common toxicity associated with CAR T therapies is cytokine release syndrome, CRS, and neurotoxicity, clinical protocols of CAR products should include the grading scale and management guideline for CRS and neurotoxicity, particularly ICANS. We also recommend that sponsors monitor a panel of cytokines at the baseline and follow-up visits, and repeat the test when a CRS is suspected. I would like to emphasize several important considerations for safety monitoring. First, we review the appropriateness and completeness of safety lab and schedules proposed in the IND protocol. Second, most CAR-T products are retrovirus vector-based gene products and meet the FDA requirement for long-term follow-up for up to 15 years. Third, long-term follow-up for up to 5 years for replication-competent retrovirus or lentivirus may be required depending on testing results during the first year. Lastly, I would like to emphasize the importance of annual development safety update report, also known as DSUR. DSUR is an important means for FDA to assess overall safety of the product development program. Important safety information includes both safety events and relevant dosing information. Safety concerns may rise if there is an unusually high incidence of grade 4 or 5 events, high incidence of DLTs, or high number of subjects who did not receive the CAR product. To minimize the number of subjects exposed to unacceptable toxicities, all early phase trial protocols should include study stopping or pausing rules. To clarify, study pausing rules are intended for temporary pause of treatment to allow thorough safety review by the Trial Safety Committee or DSMB. Sponsors should consult FDA when the study enrollment and treatment is on pause. You may have seen this chart before, which shows common meetings between FDA and sponsors. In the interest of time, I only want to emphasize that sponsors should take advantage of pre-IND meeting to reduce potential hold issues in the IND submission. We strongly recommend that sponsors address all FDA comments from the pre-IND meeting in their IND submission. This slide shows you some of the resources for meetings between FDA and sponsor. To summarize this brief review, there are at least 2 take-home messages. First, FDA's review of early phase trials focus on safety and every aspect that is pertinent to the safety of study subjects. Second, sponsors should go beyond identifying MTD in first-in-human studies and incorporate optimal dose finding strategies into early phase trial design. As I promised in the beginning, we have 2 challenge questions to test your knowledge. The first poll question is, which primary endpoints are appropriate for a first-in-human study? A, overall survival, B, safety, feasibility, dose finding, C, landmark progression-free survival rate, D, objective tumor response rate. Which one is correct? Yes, you're right. The correct answer is B. The second question is, which of the following is or are important clinical safety information? I'll give you a few seconds to answer this. Cumulative summary of SAE, the rate of DLT, number of subjects treated at MTD, number of subjects who did not receive CAR-T cells. Most of you get the correct answer. All of them are important safety information. This slide has listed useful FDA guidances, and this one too. This concludes my talk. Many thanks to your attention. Lastly, I would also like to thank all of my colleagues for their support and input.
Thank you very much for inviting us to be part of this SBIA event. I thank also the audience for joining this presentation. My name is Kassa Ayalew. I'm the Director of the Division of Clinical Compliance Evaluation in the Office of Scientific Investigations within the Center for Drug Evaluation and Research at the FDA. My presentation titled International Clinical Trials from a GCP Perspective focuses on international clinical investigations that are conducted for the purpose of collecting safety and efficacy data to support marketing approval of medical products from CDER's perspective. Disclaimer slide. The views expressed in this presentation are those of mine. They are not necessarily those of the Food and Drug Administration. The objectives of my presentation are to provide you with a general overview about international clinical trials from GCP perspective, as well as to share with you information on how, as clinical investigators, you can comply with FDA's regulatory requirements and expectation when involved with international clinical trials. Introduction to international clinical trials. As most of you know, clinical trials are increasingly global for many reasons. The simplistic diagram to the right of the text on the slide is intended to demonstrate the global nature of clinical trials that are contributing data to marketing submissions or authorization to the US FDA or to any other regulatory agency for that matter. Clinical trials in support of FDA marketing applications can be conducted under a variety of scenarios. The minority of those clinical trials may be conducted or may be based solely on foreign countries, on a single or few limited number of countries. The preference, at least by the FDA, would be to have multinational clinical trials to support marketing-related submissions. Again, as you know, the FDA has regulations regarding the conduct of clinical trials and the acceptance of data that come from international clinical trials in support of marketing applications. We'll be discussing about those relevant FDA regulations on the next slide. What are the relevant FDA regulations we just mentioned about? As clinical investigators, it is very important for you to know about the relevant FDA regulations in relation to conducting international clinical trials and about the acceptance of international clinical trial data by regulators such as FDA. Those relevant FDA regulations have objectives to assure the safety and the rights and the welfare of study participants, as well as to help assure that the quality of the scientific evaluation of the drug would be adequate to permit an evaluation of the drug's effectiveness and safety. We encourage and recommend clinical investigators such as you to learn more about the regulations by FDA governing international clinical trials. Some of the most relevant FDA's regulations that are crucial for clinical investigators to know are listed on this slide. Those highlighted or boldened are very important for our discussion today. But all of the regulations that are listed on this slide are critical and important for a clinical investigator to know when it comes to conducting international clinical studies. Again, all are important, but it would be crucial for clinical investigators to know the relevant FDA regulations that governs their involvement in conducting and producing data to support marketing application in the US. Conducting clinical trials outside the United States. As you know, IND is not required to conduct a clinical trial outside the United States. However, if a clinical trial is conducted under an IND, irrespective of the location where the study may be conducted, the study must comply with the relevant FDA regulations such as 21 CFR 312, the regulation that governs IND application, unless the requirements are waived. If a clinical trial is not conducted under IND, the sponsor must ensure that the trial complies with the requirements in the 21 CFR 312120, which is the regulation that we recently mentioned about that governs the foreign clinical studies, the acceptance of foreign clinical studies not conducted under IND. As you know, an IND is a request for FDA authorization to administer an investigational drug to humans. An approved IND application would allow investigators to proceed with a new drug trial, and the information collected from that IND may later become a part of NDA if the trial becomes successful. Outside the United States, there can be a mix of IND and non-IND sites for a study, as IND is not required outside the United States. Non-IND studies outside the United States. The FDA has detailed regulations regarding the conduct of non-IND studies and the acceptance of clinical trial data from non-IND studies. Non-IND studies should be conducted in a manner to support a marketing application according to FDA's regulatory expectations. First, non-IND studies must be conducted in accordance with good clinical practice. When studies are not conducted under IND, the sponsor must ensure those studies comply with the local regulatory requirements as well as the GCP expectation. The non-IND studies must be well designed, well conducted, and performed by qualified investigators. And secondly, FDA should be able to validate the data from non-IND studies through an on-site inspection if necessary. Foreign and domestic inspections are conducted between fiscal year 2012 through 2022. The foreign and domestic inspections conducted by FDA during the previous 10 years are shown on the slide. Most of the bioresearch monitoring related inspections for drug and biologics as shown on the map occurred in the US and several European countries. About the fifth of the inspections were done outside the United States. Stated before, it is very important for FDA to validate data through on-site inspection both for IND and non-IND studies. The FDA inspects foreign scientists, in particular if there is insufficient US or domestic data or only foreign data is submitted to support marketing applications. We may also consider to inspect foreign scientists when domestic and foreign data show conflicting results, important for regulatory decision making. The other instance that we may inspect foreign scientists would be when there are serious issues to resolve, such as suspicion of fraud, scientific misconduct, and significant human protection violations. The focus of FDA's inspections outside the United States, in particular for non-IND studies, is on ensuring that the data presented can be relied upon for regulatory decision making. In short, it is about finding whether the data presented to FDA fit for purpose. Clinical trial requirements in the United States. In the US, investigators are required to submit an IND to FDA if they intend to conduct clinical investigations unless the study meets specific regulatory exemption criteria, which we'll be discussing later. When a clinical study is conducted under an IND, all FDA IND requirements under 21 CFR 312, which is a regulation that governs an IND application, must be met unless waived. Let's look into investigator responsibilities for IND studies. As already discussed, FDA requires investigators to meet their responsibilities to ensure the rights, safety, and welfare of their study participants, and to help assure that the quality, reliability, as well as integrity of IND studies. On this slide, we will be looking into the most important responsibilities that you as clinical investigators are required to meet for IND studies. First, clinical investigators for IND studies are responsible for ensuring the investigation is conducted according to the signed investigator statement, which is also called FDA Form 1572, and are also required to conduct the study according to the investigational plan and applicable regulations. Second, clinical investigators are responsible for ensuring control of drug under their investigations and ensuring adequate precautions as well as storage of the investigational product. Thirdly, clinical investigators are responsible for maintaining adequate records of the disposition of the drug and accurate case history. Number four, clinical investigators for IND studies, you would be responsible for ensuring record keeping and retention according to the FDA's regulation. Number five, if you are involved in IND studies as clinical investigators, you would be responsible for providing investigator reports that includes progress reports, safety reports, and final study reports. Number 6, clinical investigators for IND studies. You would be responsible for ensuring IRB review, approval, and meeting the reporting requirements by IRB. Number 7, and if you are involved in IND studies as clinical investigators, you would be responsible for ensuring FDA has access to inspect investigators' records and reports. Number 8, and clinical investigators will be responsible for ensuring the protection of the rights, safety, and welfare of the subjects. Number 9, and it's very important as clinical investigators for IND studies, you would be responsible for ensuring that informed consents are obtained before patients begin the study. Number 10, as clinical investigators for IND studies, you would be required to provide financial disclosure information to the sponsor. Not meeting the most important IND responsibilities as clinical investigators could lead to issuance of Form FDA 483 citation after inspections are concluded. Therefore, in order to avoid being issued a 483, it is very important to meet the investigator responsibility for IND studies. What are the exemption criteria from IND requirements? Exemption from IND will be granted if all of the following requirements are made. First, the drug product for a planned investigation should be lawfully marketed in the United States. Secondly, the planned investigation should not be intended to support new indication or significant change in the labeling. Third, the planned investigation should not involve a route of administration or dosage level or use in a patient population or other factor that potentially increase risks to the patients. Number 4, the investigation should be conducted in compliance with Part 56 and Part 50 of the regulations that are related to IRB oversight and obtaining informed consent prior to beginning of the study. 5, the investigation should be conducted in compliance with 21 CFR 312.7, which is a regulation that governs the promotion and the commercial distribution of an investigational new drug. Acceptance of foreign data from clinical trials not conducted under IND. As already discussed, FDA accepts foreign data from non-IND studies to support marketing applications or submission here in the US. In order for the foreign data to be accepted by the Food and Drug Administration, the data should be first applicable to the US population and the medical practice. Secondly, the foreign data should be generated by investigators of recognized competence. And thirdly, the foreign data should be considered valid even without doing an inspection. Last, fourth but not least, FDA should be able to validate the data through on-site inspection or alternative mechanisms when it deems necessary. As already discussed under 21 CFR 312120, FDA will accept a well-designed, well-conducted non-IND foreign study if the study is conducted according to GCP and if it was conducted ethically. As clinical investigators, you should know that marketing approval for new drugs based solely on foreign clinical trial data is governed by the FDA regulations 21 CFR 314.106, which is a regulation that governs application for FDA approval to market a new drug. General expectation during inspection of clinical investigators. As clinical investigators, you should know that as part of medical product approval or a review of submission, the FDA regularly performs inspections of clinical investigators and their sites to ensure compliance with GCP as well as FDA regulatory requirements. FDA investigators may inspect study binders, drug accountability records, consents, medical records, and records related to tests and procedures, case report forms, qualification of investigators and study staff, as well as all other documents that may relate to the clinical investigation. Inspection areas of focus. As you all know, data validation through inspection is a very important part of marketing application review and approval. The key areas inspectors will be focusing during inspection of clinical investigators include checking for protocol adherence with a focus on the conduct of important procedures, such as making sure the procedures related to primary endpoints or important safety assessments are done properly. Second, the inspectors also will be focusing to look at the review of informed consent as well as review of higher review oversight to ensure the ethical development of a medical product. Third, inspectors will be comparing line listings, so subject line listings submitted to FDA by comparing it with source documents and verifying the reported data, both for accuracy and data quality. Fourth, inspectors will review enrollment process. They look into monitoring activities and review the reporting requirements to sponsor, as well as compliance with applicable regulation when it comes to safety reporting, financial disclosure information, and any updates related to the study. The inspectors also review test article control and records custody and retention during inspection. Well, this is the final slide. The take-home message: as clinical investigators, it is important that you know FDA's regulatory requirements for international clinical trials are crucial to reliably produce high-quality data without jeopardizing the rights, safety, or welfare of the trial participants. The knowledge about FDA's regulatory requirement for international clinical trials is also important to comply with FDA's regulatory expectations. Hope the general overview on international clinical trials from GCP perspective was helpful to you, and I would be more than happy to entertain any questions you may have at the end of this session. And thank you very much for attending this presentation.
Thank you very much to all our presenters. Some really fascinating updates on new science and new therapeutics that brought new hope to many patients from Lei and from Lianne. And thanks very much to Kassa for outlining our perspective on international trials, which is obviously crucial as such a high percentage of the trials we review are international. I'm going to take a little bit of latitude. I see that we've officially almost run out of time, but I believe I've been permitted to ask for an extra 5 to 10 minutes just to deal with some of the questions before we wrap up for the day. So without any delays, I'm going to move into a couple of questions. First of all, first question, there's a question for Lei here. Lei, how often is placebo used for control? It seems diseases using gene therapy would be serious and some treatment would be needed. Maybe we need a little bit of time for Lei. Lei?
Yes, can you hear me now?
I can.
Okay, so as I really emphasize in my talk, because most of the gene therapy trials are for rare conditions. That's why we really encourage sponsors to conduct like a randomized concurrent control study from get-go to collect the data that could provide evidence of effectiveness and safety. And for later phase trials, we have been consistently asking for randomized placebo, not placebo, but concurrent control studies for most of the conditions. I understand that for Zolgensma, which was approved based on 2 single-arm studies, but that's a very unique situation because the disease is well characterized, the natural history is well known, and is very homogeneous in that SMA1 population. And the outcome is the treatment effect is large. We know those babies will not be able to sit independently without the intervention. So because of that, and we think that single arm study is adequate and well-controlled study for the BLA approval. But in most cases, that's not the case. So that's why it is critical to conduct well-designed studies to ensure the data are interpretable, both for the safety and efficacy. And I would recommend the sponsors to really read carefully of our guidance documents on gene therapy for neurodegenerative conditions and gene therapy for retinal disorders and some other gene therapy guidances. Thank you.
Thanks very much, Lei. There's a question for Lianne here, also to some extent focused on single-arm studies. Most of the pivotal trials in cell and gene therapy were single-arm. Some received full approvals, others accelerated approval. And the question is, how does OCE decide which pathway to use for approval? Do you expect the ratio of full approvals to accelerated approvals to change over the coming years? So really a perspective on accelerated approval in presumably cell therapy. Any thoughts on that, Lianne?
Hi, this is Lianne. Not to crash, so I'm not going to use video. So this is a great question, actually. I think the audience already noticed that I mentioned in the talk that I can speak for CAR T cell, especially that in this year we proved two indications in the early line settings, and that based on the randomized control trial and regular approval. But in the past, almost all of them are based on single arm trials with very high objective response rate and very high complete response rate, and so how they determine the pathways really depend on the clinical benefit and totality of evidence. If it's single-arm trial, in a lot of cases for the CAR T therapy in the later line, and patients already received many, multiple lines of therapy and no other, viable treatment in the standard of care setting anymore. In that case, I think it's only single-arm trial may be feasible. But we do have a trend, and if CAR T cell works, it may be able to very likely look at this new approval, move into the early line therapy. In early line therapy, there are standard of care, there are comparisons, so you can do the head-to-head comparison. You can look at the time to event, especially event-free overall survival, which is the ultimate benefit. So in that case, there seems to be a trend, but I can't say this ratio is going to change, very soon. Thank you.
Thanks for an excellent answer there. A question for Kassa about international trials. When will the GCPA inspection take place? The questioner asks. For example, is this going to be after the submission of an NDA or earlier, e.g. after phase 2 or 3 trials, or even during a clinical trial? And the second part seems to be, how much time is there between the announcement of the GCP inspection and the date of inspection? A couple of procedural questions. Something that you can answer for us, Kassa? Looks like we're having trouble with Kassa's sound as well. I'm going to suggest that he turns off his video.
Now, I was unable to unmute myself, Leonard. I heard the question, and the first part is inspection throughout the clinical trial development life cycle can have. I mean, the purpose of inspection is, in addition to verifying the data reliability in the integrative study, it's also to ensure that patients who are participating in clinical trials, their rights and safety and welfare are protected. Therefore, in some cases, I mean, most of the time we do routine clinical trial data verification, in particular for applications or submissions that where there is a great interest to review and approve a product. But in some instances, we may also, because of compliance, because of concerns or triggering some integrity or patient safety. Those are, I mean, the types of inspection we do. When it comes to the timing, and in general, for routine surveillance kind of inspection, where we verify data submitted to the agency by comparing the information we have been given with the information in the source records, like at the clinical investigator site for the, in particular for this audience, and we will be providing, I mean, inspectors, our inspectors will contact clinical investigators or entities of interest, I mean, days ahead of the inspection. In general, about a week. Investigators not to in any way pay those kind of inspection that can be seen as a violation. Therefore, usually in general, it would be about a week. But I mean, usually, I think, I mean, there will be instances. There are many factors that could use pre-announcement that needs to be taken into account. In general, about a week. Leonard, thank you very much.
Thanks, Kassa. I have another one, a quick one for you, which I know we've spoken about. Is the 1572, Form 1572, required for foreign IND studies, or can it be waived?
Right, that's a great question, and that's a very important question, actually. And if intends to conduct the study under IND, 1572 would be required, right? Because if somebody is intending to conduct the study according to the US law, 1572 would be required. However, I mean, just like any other regulation or requirement that the agency has, sponsors can ask for a waiver, and they need to come with a reasonable justification, perhaps an alternative way of providing the same information that we require to ensure the conduct of reliability and force. Therefore, yes, waiver can be asked about any kind of regulatory. Not sure, Leonard, if I have answered the question.
Oh, no, I think that's a good answer. Thank you. Thanks, Kassa. Let's see, a couple more questions. I guess here's an interesting one for Lei. The question is, do you see any toxicity in gene therapies used in eye diseases?
The short answer is yes, but for the eye indications, because most of the products, if not all, are delivered locally, which most often subretinal injection, such as in the case of Luxturna, and some of them are delivered intraocularly. So I think most of the AEs or SAEs we observed have been mostly like local SAEs, the ocular SAEs. And I think if you look at the label of Luxturna, the serious ones include like cataracts formation or more speeding up of the worsening of the existing cataracts, this ocular inflammation, and those type of adverse events. Thank you.
Great, thanks very much. I'm just looking through the questions again. Yeah, perhaps one for Lianne. The question, I think, has to do with the phase of development. Does FDA approval of CAR T products require a phase 3 trial? Is that something you can talk about, Lianne?
Okay, thank you, Dr. Sachs. It's a good question. it's not like a FDA, require you must have a phase 3 trial. And usually we call pivotal trial. Pivotal trial can be phase 2, can be phase 1. It really depends. If a phase 2 trial, even is smaller, but if the evidence is so strong, so robust, and is meeting the criteria for regular approval or accelerated approval, you go. That phase 2 can be used for approval. But, it all depends on the evidence of the efficacy and also the safety.
Right, thanks very much. Yeah, I think that's great.
Thanks.
I think we're sort of at the end of our time, so perhaps just leaving time for Brenda to wrap up. I just wanted to thank our speakers very much. It's been a great session and really very informative in a number of different ways. And of course, I wanted to thank our enthusiastic audience for all their participation questions. And just wanted to remind you that tomorrow we'll be picking up on a couple of different themes. We'll be talking about compliance, we'll be talking about novel trial approaches, and we will be talking about the investigator's brochure, non-clinical studies. So please join us for those.